public display part of the museum out front is what most of the public sees about our museum and any museum, and that's what most people think a museum is a bunch of mounted displays that are for public education, and the public education part of a museum is a very important part, but it's not the only thing. And really, for our museum and for most museums, the public displays are the tip of the iceberg, and what most of the museum is a research collection. People are looking at these skins and they're looking for those subtle differences between populations. What's become a lot more common recently is, with the advent of sort of the genetics revolution, is to do these same sorts of things, but with other sorts of markers that we can get from the skins that were not obtainable, a few decades ago. One of the biggest things right now is genetic markers.
The non-traditional uses of a museum that involves a really modern technique that has kind of exploded in use in recent years is what's called the stable isotope technique. The atoms that are used in living things like nitrogen and oxygen and carbon, they have different isotopes that have different atomic weights. And when an animal eats something, nitrogen is what is in amino acids and proteins, and the nitrogen gets metabolized, it gets deposited in different forms of protein, like, for example, feathers or hair. And it turns out that the concentration of the different isotopes, say nitrogen 14 versus nitrogen 15, depends on where the animal is in the food chain. Here we have some marbled murrelets, which is a small seabird. And there's this fellow, Ryan Norris, in Canada. Right now he's at Guelph University in Canada. And what he was interested in was whether or not marbled murrelet diets had changed over the centuries because of overfishing. His theory was that because of fishing, the fish in the sea that the marbled murrelets have to eat are smaller and at a lower trophic level, or by trophic level I mean they're lower in the food chain. And so what he wanted to do was to look at stable isotopes in the feathers of murrelets over a century and see if he could detect any differences. So he asked everybody who had marbled murrelets in their museums to take little bits of feather, and the thing about stable isotopes is all you need is just the tiniest little bit of a feather, so you can't see that we took any samples from these. He did find that marbled murrelet diets have changed over the past century, probably due to overfishing. When you look at a chipmunk out in the wild, they're moving really fast. They're little tiny thick creatures, and one of the advantages of having museum specimens is that you can actually see the differences.
So you can see for these chipmunks here, these guys over here are slightly redder and slightly bigger than these guys over here, suggesting that maybe they should be different species. And now with these modern tools, we can also look at the genetics to see how genetically different they are from each other. And then this is one of the birds that I study. This is an Australian fairy wren. And again, we're interested in the evolutionary history of these birds and are able to obtain genetic material from the study skins. The question that we're interested in is the speciation process itself. How do you get one species splitting into two? And the genetic samples that we collect from these museum skins are helping us do that kind of work.
You can also, in historic sites, get to socioeconomic status by looking at the bones they use. There's certain cuts of meat that are higher status. I did a project earlier, Butte, Chinatown, where I had ethnic Chinese butchering practices versus dominant Euro-American butchering practices. And sometimes you can chart how much the population is being absorbed into the wider culture by looking at just the butchering, the bones they use, the meat cuts they use. I could not do this job without this museum. This is essential, having a comparative collection of this caliber, especially the bird collection. there's just no way a single individual can collect enough bones to do a good job. There are written keys, but it's not the same as taking a bone like the head of this femur and then looking at the head of the femur of a sheep or a goat and saying, okay, that's what it is.
It's a big investment in the future, and it takes a lot of effort. I mean, it takes a lot of time and space to hold the collection and things like that. But yeah, it's an investment for future research.